Hi everyone. Welcome to the second in this UK DRI research webinar series, Rethinking Neurodegeneration. Before we start, I just want to give some background to the UK DRI. Next slide, please. The UK Dementia Research Institute was founded in 2017 by our principal funder, the Medical Research Council, together with the two dementia charities, Alzheimer's Society and Alzheimer's Research UK. Since then, we are now lucky enough to receive support from a number of organisations, including LifeArc, NIHR, the British Heart Foundation, and Parkinson's UK, the latter two of which have allowed us to set up new centres focused on vascular dementia and Parkinson's research. In a rapidly aging society, our vision for the future is healthy brain aging for all. Next slide, please. At the heart of the UK DRI's work is the unpicking of the underlying pathobiology of a range of conditions that lead to neurodegeneration. This is based on the principle that better mechanistic understanding of the causes and drivers of disease underpins the development of new diagnostics, technologies, and treatments that have the potential to deliver life-changing solutions for people affected and at risk. Since being founded in 2017, the UK DRI has grown to 9 research centres across leading UK universities, hosting 85 labs of group and emerging leaders, and made up of over 1,000 researchers, clinicians, students, and support staff working on this single mission to transform the lives of people affected by or at risk of dementia and other related neurodegenerative conditions. Next slide, please. And so we come to today's talk. Before I introduce Marc, I just have a couple of housekeeping things to cover. Firstly, to let you know that this session is being recorded. And secondly, I'd like to encourage you all to please put your questions in the Q&A box throughout the talk, and we'll try to address as many as possible after the presentation. Our speaker today is Dr. Marc Aurel Busche, a psychiatrist and neuroscientist at the UK Dementia Research Institute at UCL. Marc attended medical school at the Ludwig Maximilians University of Munich and completed his clinical training in psychiatry and psychotherapy at the University Hospital of the Technical University of Munich. His major research focus is to understand the cellular and circuit mechanisms that underlie the initiation and progression of Alzheimer's disease-related disorders, so that, through this understanding, more effective therapies can be developed. He also provides patient care as an honorary consultant psychiatrist in the Cognitive Disorders Clinic at the Queen Square National Hospital for Neurology and Neurosurgery. He is the recipient of several awards and fellowships, including a UKRI Future Leaders Fellowship. My name is Dr. Rajani, and I'll be chairing today's session. I'm a group leader at the BHF UK DRI Centre for Vascular Dementia Research based at the University of Edinburgh, and my group focuses on understanding the causes of white matter damage in vascular dementia. I also previously worked in Marc's lab as a senior research fellow, working to understand the role of oligodendrocytes in Alzheimer's disease. Today, Marc will be discussing his recent publications and sharing his insights on how brain circuits fail in early Alzheimer's disease. Over to you, Marc.
Thank you so much, Rakesh, for the introduction. I'm delighted to be part of this new seminar series on rethinking neurodegeneration. And what I'd like to do today is to share my views and data that support a slightly novel perspective on neurodegenerative disease, one that takes into consideration more how the brain works and brain circuits in Alzheimer's disease and related disorders. And the key questions that I'd like to address are: how do brain circuits fail in early Alzheimer's disease? That's also the title of today's talk. But an equally important question, I think, is how can we make circuits and the brain more resilient to the effects of pathology? It's becoming increasingly clear that often in neurodegenerative diseases, multiple pathologies are present. And one way to address this is by addressing all these different pathologies. But an alternative way might be to make brains more resilient to multiple pathologies, as the brain is able to cope, at least for many years to decades sometimes, with pathology. We just need to extend this slightly, and I'll make some suggestions how we could address this during my talk. So this is the standard model of neurodegenerative disease. Proteins accumulate and aggregate in the brain. That's why these diseases are often called proteinopathies, which leads to region-selective neuron loss, and ultimately dementia. And in Alzheimer's disease, which is the main topic of my talk today, two proteins accumulate, amyloid beta and tau. Amyloid beta accumulates in form of amyloid plaques that are extracellular and are believed to initiate the disease. Tau accumulates in form of neurofibrillary tangles inside the neurons, and again, this leads to region-selective neuron loss and ultimately cognitive decline and dementia. And this so-called amyloid cascade model has been highly influential and successful and has led to the first disease-modifying therapies for Alzheimer's disease targeting the amyloid component that initiates the disease. Nevertheless, there are also some fundamental limitations to this model which cannot explain certain aspects of disease initiation and progression. And in a way, it also limits us to a number of targets, in particular Abeta and tau. There's some observations that challenge the model, and this is by no means a complete list here. But for example, we now understand that it takes up to 30 years from the first development of plaques to dementia. Also, tangles and nerve cell loss can occur in the disease many years before cognitive symptoms or impairment occur. And to me, this suggests that while pathology might be necessary, it might not be sufficient to cause clinical symptoms and disease progression. And we need to consider more the circuitry in which the molecular pathology accumulates over many years. There's enormous heterogeneity in the onset and rate of progression in the disease. Some people start having symptoms as early as in their 40s or 50s, others in their 90s or later. And also the rate of progression can vary dramatically. Often astonished to see that similar levels of atrophy or similar levels of pathology can be associated with very different rates of progression or ages of onset. Pathology and atrophy do not match. We know, for example, that the number of cells lost in Alzheimer's disease is far greater than the number of tangles, which are often correlated to atrophy, and also cognitive behavioral symptoms do not correlate well with atrophy. For example, in early onset cases, very often the MRI scan looks normal, although symptoms are already present. And in other neurodegenerative syndromes, for example, semantic dementia, atrophy can be quite advanced despite relatively moderate symptoms and relatively little functional decline. So our working hypothesis is that there's a greater role of a dysregulation of neural circuits, and I use that term now in a very broad sense, and I will try to unravel this during my talk a bit more. And of course, sometimes people put neuronal dysfunction or synaptic dysfunction somewhere here in this linear model, but I think that doesn't capture the complexity and importance of the role of circuitry changes, in particular in the beginning, in the interaction with the Abeta and tau accumulation, but also later in the disease in relation to cognitive symptoms, which cannot be alone explained by the loss of neurons or synapses. So the two underlying questions we're trying to address today is which circuits and cell types show the earliest and strongest susceptibility to pathology, and how can we at least begin to address to boost circuit resilience to protect against pathology. So how can we make circuits more resilient? Now, the tools that we're using, I consider basically the model systems or the systems that we're using, the technology and the readouts. Of course, we're dealing with a human disease that unfolds over many decades, and this is very difficult to capture in the laboratory. And we can use model systems that recapitulate certain aspects of the disease. So for example, mice engineered to develop amyloid plaques, and then can ask questions about how these plaques affect the surrounding nervous tissue, for example. In humans, we can study circuits using technologies like functional magnetic resonance imaging, EEG, MEG, which are very powerful and tell us something about how the brain works. But the limitation is that they don't allow a single cell resolution. And ultimately, we'd like to understand the disease at the single cell level, which we can do in model systems. And I'm going to talk about techniques like patch clamp recordings, which allow us to record from a single neuron the activity, two-photon calcium imaging, which is a technique that allows us to record from populations and larger numbers of neurons at the same time. And a newer technology is called high-density neuropixels, which allows us to record from multiple brain regions at the same time. And the readouts we're interested in are essentially spikes or action potentials and oscillations, which are highly conserved across species, and therefore we think they have some translational value. Just wanted to illustrate this point with this slide. I'm not going into too much detail, but essentially this shows an intracranial recording from a human during surgery and an intracranial recording from a mouse. And I hope you can appreciate that the signals we're dealing with in these two systems are very similar. So we are confident that the types of readouts we're looking at have some translational value, and we're trying, of course, to always anchor findings in mouse studies, which is the majority of findings I'm going to present in human data. Now, another very important and interesting aspects about the brain is that it is constantly active. The neurons are constantly active during rest, during sleep. It's one of the major features of the intact brain in vivo. And here on the left side, you will see an example two-photon calcium imaging recording. The neurons are labeled with a fluorescent calcium indicator. And every time a neuron lights up, this means it fires an action potential, a spike. You see how active the neuron is, the brain is during rest, but also the very high spatial resolution that two-photon calcium imaging can provide. And then on the right side, you see a typical raster plot from a neuropixels recording. Here, each row is a neuron and each dot is an action potential. And because the shank of these recording electrodes is long, we can capture activity from multiple brain regions. So here, for example, cortex, hippocampus, and subcortical structures. And this spontaneous activity is well understood and is important and gives us a good readout of overall synaptic connectivity and drive in a system. It's almost like a canary in a coal mine. If something goes wrong at this level of spontaneous activity, it's very likely that higher order activities and networks are impaired as well. Another aspect why spontaneous activity is, in our view, important, because we recently discovered that the amyloid precursor protein family, the APP family, is important in regulating and maintaining this spontaneous activity. We showed that when we depleted neurons of the APP family, protein family, they fail to generate or are unable to generate this kind of spontaneous activity anymore. And many neurons are functionally silent in the brain when the APP family is absent. So I think it's very interesting that the APP family, which we always consider in the context of amyloid formation as the precursor of amyloid beta, physiologically plays an important role in maintaining spontaneous brain activity. And in that paper, we also showed that this is due to an interaction with NMDA receptors. So by it stabilizes NMDA receptors at the synapse, and when the APP family is absent, these NMDA receptors are reduced and the synaptic glutamatergic drive is reduced. And I think it's important to consider this when we're talking about the effects of Abeta and APP derived proteins on the brain. Now, this is another example of two-photon calcium imaging where we looked at this spontaneous activity and asked the question, how is it impacted by the presence of Abeta plaques in mice that produce these amyloid plaques here in the cortex? And in these experiments, we discovered that many neurons in vicinity of these plaques become abnormally hyperactive, here shown in red. And we mapped these neurons across the entire cortex and found that most of these hyperactive neurons were actually clustered in very close proximity of the amyloid plaques. So we hypothesized that removing plaques would ameliorate or rescue this abnormal hyperactivity phenotype of the neurons. And we did this experiment by treating the mice with a monoclonal antibody against Abeta 3D6, and we could show that this antibody indeed reduced the number of plaques in the brain compared to a control group. However, in contrast to our expectation, this hyperactivity phenotype was not rescued, it was further exacerbated. So instead of 20% or so of the neurons being hyperactive, now over 50% of the neurons were hyperactive. And we think that could have explained the lack of clinical efficacy of these first generation monoclonal antibodies that were in clinical trials. 3D6 is the precursor to bapineuzumab. But perhaps more importantly, it also led to an insight, namely that perhaps not the plaque per se, but the soluble species of Abeta are more important in promoting this abnormal hyperactivity effect. And indeed, we showed that taking soluble Abeta and administering it to neurons in healthy wild-type mice relatively acutely elicits hyperactivity of neurons. And this effect was confirmed by a colleague of mine in Munich a couple of years later, who used actual soluble Abeta derived from human brains and sees the same effects. And we now know that the current generation of monoclonal antibodies that are approved are much better in capturing these soluble species of Abeta and thereby also reduce, of course, plaque levels relatively quickly. Now, there's another important aspect, which is brain region-specific vulnerability. All these recordings I showed were in the cortex, and we were very interested to understand what happens in the hippocampus, which is important for learning and memory. And we performed 2-photon calcium imaging, again, now in the hippocampus, not in the cortex, and we discovered that at a very young age in these mice, so 1.8 months, at this time they didn't even have plaques. So this is a stage in humans that would be undetectable, because the first thing we can detect is probably plaques. So at a relatively young age, we already see the hyperactivity of these hippocampal neurons at a time when the cortex was still normal, at least in the superficial layers. So that suggests that the effects of Abeta, the effects of soluble Abeta, also depends on the brain region, and the neurons in different brain regions react slightly differently to Abeta. Now, this is not a one-way street. We think this is bidirectional, and this is work from David Holzman's group and others who show that when one stimulates neurons and then measures the Abeta levels in the interstitial fluid, so the fluid that surrounds the neurons, Abeta increases. And when one blocks neuronal activity, this does not happen. So these experiments have led to a concept in the field in which neurons are the primary producers of amyloid beta, and in Alzheimer's disease, amyloid beta accumulates. We're agnostic about why it accumulates. It could be overproduction in familial cases, or it could be impaired clearance. But when Abeta accumulates, it triggers neuronal hyperactivity, and this hyperactivity then can in turn lead to more neuronal release. And my group and many others have operated under these assumption until Rikesh, whom you briefly met in the beginning, joined the laboratory and looked at gene expression across multiple cell types and multiple data sets and discovered that the machinery and the genes that are important for Abeta generation, APP, base 1, presenilin 1, are highly expressed not only in neurons, but also in oligodendrocytes, so the cells that produce the myelin or myelinate neurons in the brain. And these are heat maps, so red means more expression. So Rikesh wanted to confirm this in tissue and showed that indeed the majority of the oligodendrocytes, so over 80% or so, contain the machinery to produce A beta. And interestingly, in tissue from Alzheimer's patient, the number of these oligodendrocytes was increased. So in the Alzheimer's tissue, there seems to be more oligodendrocytes that could potentially produce A beta. Now, it's very difficult to measure A beta in tissue. Rikesh established a cell culture system using human-derived iPSC oligodendrocytes, which produce myelin. And then he collected media and measured the levels of A beta in that media and compared it to other cell types. And what he found was very interesting, namely the oligodendrocyte cultures had most A beta release within 24 hours compared to neurons and also other cell types. So it seems that in the brain, at least under these conditions, oligodendrocytes and neurons produce most of the A beta. And that was quite surprising. So we wanted to validate this finding, and we collaborated with David Klennerman, who's a group leader at the UK DRI in Cambridge and a pioneer in super resolution methods to image very small nanometer scale protein aggregates, including A beta aggregates. And his team confirmed that oligodendrocyte media contained 3 times the amount of A beta aggregates compared to the neurons. So we asked what the functional consequence is of oligodendrocyte A beta production. And here we again turned to neuropixels recordings and looked at a different amyloid model, APP and LGF mice, and confirmed first that they display hyperactivity of neurons in the cortex. And then we compared them to a model in which BACE1, one of the enzymes that's important for A beta production, was selectively knocked out in oligodendrocytes. So the oligodendrocytes could not produce A beta anymore. And to our surprise, this completely rescued this hyperactivity phenotype. And I say to our surprise because in these mice, the neuronal A beta is still produced and around. So this suggests that either the oligodendrocyte A beta is more bioactive or has specific characteristics that makes them more toxic, or there's some kind of threshold effect that could explain this. We also confirmed this by taking the media and injecting it, administering it directly to neurons in healthy wild-type mice. And as you can see, this also elicited hyperactivity. And we could show that this is A beta related because when we immunodepleted the media or treated them with a BACE inhibitor, we did not see this effect because there's a lot of things in the oligodendrocyte media that could promote this. So this suggests that our initial model of disease initiation is incomplete, and that we need to take into consideration the possible role of oligodendrocytes. We don't know yet whether the A beta from neurons and oligodendrocytes has the same effect or whether the cellular source of A beta matters for specific phenotypes. We also don't know what the drivers are of oligodendrocyte A beta formation and these feedback loops. We do know that myelination can be activity dependent. So one wonders whether neuronal hyperactivity has an influence on oligodendrocytes and potentially also the formation of A beta or the production of A beta within oligodendrocytes. So these are all very interesting open question in my view. I wanted to briefly highlight this preprint by Mina Rüthen's group, who is the center director of the UK DRI in Cambridge, where they essentially confirmed that oligodendrocytes can make A beta, and they discovered a novel pathway of A beta formation, specifically in oligodendrocytes, which does not require BACE1. And I think that's very interesting therapeutically, because BACE1 is a difficult target Um, and perhaps one can leverage these new insights to specifically inhibit A beta production in oligodendrocytes, for example, via this novel pathway. Now, an important clinical observation about A beta is that, and tau, is that there's a spatial temporal separation. A beta initially accumulates in the cortex, and tau accumulates in the neocortex. Apologies, and tau accumulates in the entorhinal cortex. So at age 65, roughly 20% of the population have A beta plaques in the cortex. Almost everyone at that time has a little bit of tau in the entorhinal cortex. An important feature of the cortex is that it has layers, and we often consider the cortex as one, but it has these very distinct layers that have certain functions. So we very simplified know that the superficial layers 2 and 3 are more important for computation, and the deeper layers 5 and 6 also communicate with other cortical areas, but they also communicate to subcortical areas. So very specific functions of individual layers, and most of the work on hyperactivity in the cortex we have done and others was in layers 2, 3, because it's easy to access with microscopy. And we have extrapolated from here across other layers. However, the deeper layers are interesting also because there's more myelination in the deeper layers, and also we know from reports that the new anti-amyloid antibodies, in particular lecanemab, seem to bind more to the deeper layers rather than the superficial layers. So we wanted to know what happens in these deeper layers. And the first question we ask is there selective vulnerability at the level of plaque distribution? But we couldn't find that plaques are specifically clustered in a certain layer in the mouse models, and that also aligns with the human literature, which does not show a clear preference for certain layers with regard to plaque formation. However, interestingly, when we then perform two-photon calcium imaging in the deeper layers, in particular here in layer 5, we found these abnormal, very long calcium transients in the deeper layers in 2 separate mouse models of amyloid formation, which we didn't see in the wild-type mice, and we didn't see in more superficial layers, and which suggested that perhaps the spike timing or something is abnormal in these mice already at a very young age, 3 months of age. This is before they actually produce and deposit any plaques. So we wanted to understand the electrophysiological basis of this better, and performed neuropixels recordings, which gives us a direct readout of spiking and also allows us to cluster neurons into excitatory neurons and inhibitory neurons, in particular fast spiking interneurons, which are primarily parvalbumin positive interneurons. And what we found is that in the deeper layers here, 5 and 6 together, there was a suppression of activity, of spontaneous activity, specifically in these fast spiking interneurons, which we could not find in the superficial layers, and also the excitatory neurons were normal at this time. So that suggested that in response to the initial amyloid formation in the cortex, there is a layer-specific effect, with the deeper layers being more affected, but also a cell type specific effect, with the parvalbumin fast spiking interneurons being affected first. We also look beyond spontaneous activity, and here in the visual cortex we could display visual stimuli, and neurons respond to these visual stimuli. For example, in this experiment we presented moving gratings in certain orientations and directions. And what we found was that the orientation and direction selectivity was abnormally increased, again, only in the deeper layers in the fast spiking interneurons. So it seems they are more tuned to specific orientations and directions. And this is unusual because in the normal healthy brain, these interneurons would be very broadly tuned. However, there's also some effects on excitatory neurons, in particular an increase in the burstiness of the neurons. So bursting here means that spikes are produced in very rapid sequence, and some data suggest that impaired inhibition can increase the burstiness of excitatory neurons. Now, what is the molecular mechanism or underpinning of these changes? One important pathway that regulates parvalbumin interneuron function is the NPTX2 GluA4 pathway. NPTX2 is produced by excitatory neurons and is released, and it binds to the AMPA receptor subunit GluA4 on parvalbumin interneurons and strengthens synapses. This is a very established neurobiological mechanism that strengthens the function of parvalbumin interneurons. And what we found was that NPTX2 was selectively reduced in the deeper layers in excitatory neurons only, as was GluA4 in the parvalbumin interneurons. Again, only in the deeper layers 5 and 6 at this very early time point. And that resulted in a reduction of excitatory synapses onto these parvalbumin somata. And I should emphasize that this was really important to consider the layer-specific effects, because bulk measures like Western blots could not show this difference in NPTX2 and GluA4 at this very early stage. Nevertheless, NPTX2 is particularly interesting in the context of human Alzheimer's disease because several studies have shown that it is consistently suppressed in people with Alzheimer's disease in the CSF or plasma, and it seems to correlate with the degree of cognitive impairment. However, there's also a correlation with reduced NPTX2. It seems to have some in people who are not cognitively affected but have pathology in their brain, and it seems to have some positive predictive value for the development of cognitive symptoms. There's translationally a very important and interesting protein. We think that this could be a starting mechanism here that we have uncovered, that the problem starts in the deeper layers. Now, then, of course, the question is, can this be rescued? And what we did in this experiment, we performed gene therapy. So we applied NPTX2 to excitatory neurons and increased the levels. And indeed, this increased the number of excitatory synapses on parvalbumin somata and also strengthened and increased the firing of the parvalbumin or the fast spiking interneurons in the deeper layers 5 and 6. So there has been, of course, many other studies that implicated parvalbumin interneuron dysfunction in Alzheimer's disease. And we believe that potentially the dysfunction starts in the deeper layers, but then as the disease progresses, it progresses also into other layers of the cortex and other brain regions. And parvalbumin interneuron dysfunction, or GABAergic dysfunction of GABAergic inhibition has been shown to be important for the tonic neuronal hyperactivity phenotype that I spoke about initially. There's a reduction in GABAergic synapses around plaques, and we had shown that strengthening GABAergic inhibition reduces the number of hyperactive neurons. It has been linked to the occurrence of epileptiform activity and seizures in Alzheimer's disease that affects up to 40% of, or can affect up to 40% of the patients, and these seizures are often subclinical. It has been linked to impaired gamma oscillations, which are important for cognition and cognitive operations. And we have also shown that the Abeta dependent impairment of slow wave oscillations during sleep, which are important for memory consolidation, can be rescued by strengthening GABAergic inhibition. So it seems that a dysfunction of parvalbumin interneurons is really at the core, and there are multiple phenotypes in the disease that occur during the progression that can be traced back to this dysfunction of parvalbumin interneurons. Now, in the last part of my talk, I'd like to, before I put it together in a concept, I'd like to speak about tau, because it's more correlated to cognitive decline, which I think is nicely illustrated in this example. This is someone on top here with a normal cognitive function, and at the bottom, someone who has a mild cognitive impairment, and both have the same levels of amyloid plaques in the brain based on amyloid PET imaging. The main difference is that the person with mild cognitive impairment has tau accumulation in the medial temporal lobe, which also predicts later atrophy in the same areas. Although, as I said before, this the number of neurons lost is far greater than the number of tangles in the brain. So I think the tangles here are a marker of tau pathology. It's not necessarily, the tangles are not necessarily the cause of the neurons lost. In fact, some evidence from Brett Hyman's lab shows that the neuron that contains a tangle survives, and the neuron that doesn't have a tangle is the one that dies, and this could be due to more toxic soluble species of tau. Indeed, tau is an axonal protein which stabilizes the microtubules in the axon, but in Alzheimer's disease it can mislocalize from the axon, and then it can form neurofibrillary tangles, which is the hallmark of the disease, but it can also form these soluble oligomeric tau species. And the problem is very similar to Abeta. They're not as well defined as the aggregates. They're difficult to detect, and we can't really see them easily. If we look at the brains biochemically, most of the tau in the brain is this low molecular weight tau monomers, and a number of years ago, Brett Hyman's group showed that there's a rare species of what they called high molecular weight tau, which is less than 1% or so of all the tau, which is essentially the soluble oligomeric tau species, which is present in the Alzheimer's brains, but cannot be found in controls. And this high molecular weight species is interesting because it correlates directly with disease progression in individuals with Alzheimer's disease. And we wanted to understand the mechanism of this effect better. And what we wanted to do is we wanted to administer this high molecular weight tau taken from a human Alzheimer's brain and administer it to a single neuron and then observe how it affects the activity of these neurons. So what we did is we put the tau into a patch pipette, and then we patched in mouse slices CA1 hippocampal neurons so that the tau could diffuse into the cell, and then we could monitor the activity of the cell, of the neuron, over time. So in these typical experiment, what we do is we inject current into the neuron, and then the neuron responds with action potential firing. And the pattern of action potential firing is important. So the neuron can respond with a single action potential, or it can respond with a sequence of action potentials fired in rapid sequence, which are called complex spike bursts. And these complex spike bursts are essential because they can more efficiently transmit signals that support learning, memory, and network synchrony. So sometimes they're also called as a cellular correlate of learning and memory. And there's evidence that interfering with burst firing can impair learning and memory. So what happens is that in control conditions, when we apply higher currents, the neurons start to produce these bursts. However, a neuron that contains only 10 nanomolar of this high molecular weight tau, which is we think, pathophysiological relevant concentrations, so a concentration that would be present in a neuron in an Alzheimer's brain, the neurons lose the ability to respond with bursts at these higher frequencies. So it's not that the neurons can't produce any spikes anymore, because the likelihood that they respond to current injections with a single spike is relatively high. So they do produce spikes, but they cannot produce bursts. And this is because the mechanisms of bursts are different to the mechanisms of single spikes. We also confirmed that this phenotype is present in mice that express tau and accumulate tau. Here we performed in vivo patch clamp recordings of CA1 and found essentially the same phenotype, a reduction in bursts, also a reduction in the complexity of bursts. And we could link this phenotype to a reduction in the R-type calcium channel CAF 2.3 in these mice. We know from previous work that in vivo complex spike bursts heavily depend on NMDA receptors and this R-type calcium channel CAF 2.3. An antagonism of NMDA receptors or CAF 2.3 could abolish bursts. So we think that this reduction in CAF 2.3 could explain the reduction in burst firing in the context of high molecular weight tau accumulation. Indeed, when we suppressed tau in these mice, CAF 2.3 increased, and also the number of bursts in these treated mice was increased compared to a control group that in which tau was not suppressed. And this gives me a reason for optimism that even at later stages, when tau is present and accumulates in neurons, that if we can target these toxic species of tau sufficiently, then we could recover some of the neuronal function in these neurons. And we specifically showed in the paper that the effect or that there's a specific effect on high molecular weight tau. So the treatment reduced both high molecular weight tau and low molecular weight tau, but the relative effect on high molecular weight tau was actually greater by this treatment in these mice. Now, I wanted to come back to the initial model that I introduced and unravel this a bit at the end of my presentation. So we think that there's enough evidence to support a key role for Abeta in initiating Alzheimer's disease, and Abeta comes from neurons and oligodendrocytes. And a primary defect, neuronal defect or response to Abeta is an impairment in the firing of parvalbumin interneurons, which are important determinants of the overall excitability within a circuit, within a network. And the net effect is hyperexcitability, hyperactivity in the form of tonic hyperactivity, as I showed you in the beginning of my talk, in the form of hypersynchrony, impaired oscillations, and so on. So the main effect of Abeta, we think, is not an effect on cognition per se, but it changes the circuitry to a state in which the accumulation and the propagation of tau from the entorhinal cortex into other areas becomes more likely. Indeed, there is data suggesting and supporting this view that the spreading of tau across circuits is activity dependent. And once this tau, and here specifically the high molecular weight tau, has spread into the hippocampus, into the cortex, it certainly contributes to the loss of neurons, but it also contributes to functional impairments of neurons, including deficits in burst firing, which are so important for learning and memory and other cognitive operations. So we think the effect we think that there's a combined effect probably of nerve cell loss, but also functional impairments that contribute to the onset of symptoms and cognitive decline. So in addition, we know that also, and I haven't spoken about this. This is not something that we investigate a lot in the laboratory, but I think it's very important because a lot of evidence also points, of course, to microglia and how they respond to A beta very early on, and also interesting literature suggesting that microglial activation can contribute to the formation of high molecular weight tau and the spreading of tau across circuits. So based on this model that we have, A beta and tau remain very important targets, but there's also additional targets now that we think are important. And one of the key targets for me is or are the parvalbumin interneurons, which seem to determine the state of the circuit initially, and thereby produce this response by tau, which ultimately leads to dementia. So a key goal in order to boost the resilience of circuits should be to boost the function of parvalbumin interneurons. And this is again not a complete list. This is mostly human evidence. I'm not going into too much detail, but I think there's a consistent picture here suggesting that relatively low levels of NPTX2 in humans, which again is a marker of interneuron function, is associated with cognitive decline and risk to develop cognitive decline, whereas higher levels of NPTX2 seem to be protective. There's also literature or transcriptomics literature suggesting that a reduction in interneurons, not only parvalbumin interneurons, also other interneuron types, is related to worse cognitive outcomes, and high levels of interneurons, including parvalbumin interneurons, associated with what one could call cognitive resilience. And I think this really strengthens the case to focus on strengthening parvalbumin interneurons, but it needs to happen very early on and not too late in the disease. And also, lastly, what I find interesting is some literature suggesting that a lot of the evidence for cognitive or resilience and protection against cognitive decline seems to be within the deeper layers of the cortex, so layers 4, 5 to 6, which potentially matches our findings of a selective vulnerability of the deeper layers in response to amyloid beta. With that, I'd like to conclude my presentation. I'd like to thank the team who performed the work, in particular Sam Harris, who led the work on APP physiological functions and high molecular weight tau, Amalia Papanicolaou and David Grajkowski, who led the work on deep layer imaging and NPTX2, and of course also Rikesh, who led the oligodendrocyte work in the lab, and everyone else who contributed to the papers and to the work. And of course, the UK Dementia Research Institute for providing an environment where this kind of research is possible and supported, and also the other funders who support us with this work and these experiments. And I thank you for your attention, and I'm looking forward to discuss this more in the Q&A section.
Thanks, Mark, for a great talk. We've already got quite a few questions in, and please keep them coming, everyone. So the first question we have is from Emma Phillips. What makes BACE1 a difficult target for therapies?
So BACE1 has been tried in clinical trials in Alzheimer's disease, and it has not changed the disease trajectory in people with Alzheimer's disease. As far as I know, it has also led to an increase in side effects, cognitive side effects, which were presumably transient. Um, and there's just a number of targets and in particular substrates that base 1 has, including, for example, seizure 6, which has been linked to the health of synapses. So blocking base 1 or inhibiting base 1 could have potentially side effects that yeah, lead to unwanted effects. Therefore, I think base 1 is a difficult target, similar to gamma secretase, which has been a difficult target in clinical trials.
Thanks. The next question we have is from David Atwell. Perhaps I missed it, but did you say whether it is oligodendrocytes around excitatory neurons that generate A beta, or is it oligodendrocytes around inhibitory neurons, or both, or don't you know?
I think, Enriquez, you need to correct me. The answer is we don't know um, where these oligodendrocytes sit spatially. Is that, is
That's
that
right.
correct?
Yeah.
Yeah. That's a very
Next,
interesting question.
yeah, I think especially in the context of the parvalbumin data.
Exactly.
The next question is, do you think NPTX2 is always protective in neurons, or could it also be pathological if its normal interaction with complement like C1q, which has roles in pruning synapses, becomes dysregulated?
Yeah, so NPT, I mean, also have to, I mean, in Parkinson's disease, for example, NPTX2 is sky high in the striatum, and I think it's not entirely clear why that is the case. It could be because it's an activity dependent or activity regulated protein that it is very high. NPTX2 in the periphery is also, I think, linked to cancer. So it really depends on the disease. But at least from my reading of the literature and our work in Alzheimer's disease, a low level of NPTX2 suggests an impairment of parvalbumin interneurons, and therefore supplementing it or increasing it would be beneficial. Although I don't know if NPTX2 per se is the best target. That's something we need to address and understand better.
The next question is from Emma Phillips. What could be the reasons for the differences in effect seen between higher and low molecular weight tau?
So, I mean, low molecular weight tau are the monomers. High molecular weight tau is more of an aggregated tau. Um, I don't know what the biophysical differences uh, here are that are important or that are relevant. I think that's something to study more and to understand better.
Thanks. Have a question from Nina Rechorzek. Thank you for an excellent talk, Mark. Your data suggests that tau suppression had no impact on somatic CAV in 3 out of 6 mutant mice. Can you share any insights on why you think that is and what that could mean for tau targeting therapies in clinical trials? Many thanks.
Yeah, I mean, there's always variability in these mice, and in particular also to treatment responses. What I haven't shown, what I think is very interesting, and it's in the paper. There's a very clear correlation between the degree of how much high molecular weight tau was suppressed in these mice and the recovery of burst firing or the overall burst firing in these mice. So there's a very clear and significant correlation. Why some mice respond more to others, I don't know. I mean, this is a variability we observe consistently across model systems, across treatments, but it's certainly important.
And just skipping down a little because there's another question about CAV 2.3 from Jacqueline van Vierbergen. Very nice talk. How did you end up focusing on CAV 2.3 instead of other targets involved in CA1 burst firing?
So our reading of the literature was that for the complex spike bursts in vivo in CA1, NMDA and CAF2.3 are critical. So previous work from, for example, Christine Greenberger has shown if one uses NMDA antagonists or CAF2.3 antagonists, one could completely abolish burst firing. So we looked at NMDA and CAF2.3 initially as our major targets, and we couldn't see any changes in NMDA receptors, which is also in the paper, but we saw this reduction in CAF2.3. We have not explored alternative or other potential targets.
We have a question here from Ariel Krejcik. Have you looked at any alterations to perineural nets in PEV-positive interneurons in your models of Alzheimer's?
No, we haven't looked at that.
Next question from Guillaume Benora Chabot. Hello, thank you for this very interesting talk. Did you investigate if increasing NPTX2 also rescues behavioral deficits such as memory and emotional perturbations in those mice, which would provide a causal link between NPTX2 and behavior?
Yeah, this is an excellent question. Again, we haven't, but this is certainly something we'd like to explore. Does NPTX2 supplementation protect against or reduce or prevent pathology? And B, does it also protect against cognitive impairment in the mice? It's something we'd like to do, but we haven't done this.
I have a question from Larissa Fisher. Great talk, thanks. Could hippocampal deeper layers be more vulnerable to AD pathology, like you saw for neocortical areas as well?
Yeah, so I'm not entirely sure. I mean, I think there's some literature that the pyramidal cell layer in CA1 has more of a superficial and deeper component, although I'm certainly not an expert in this. And when we looked at the hippocampal pyramidal layers, we have not distinguished the depths. So I don't know is the answer to this, but certainly worthwhile to explore whether there's more vulnerability in the deeper or more in the superficial part of the pyramidal cell layer.
A question here from Dilek Merkan. How can one target high molecular weight tau for therapy?
Yeah, so importantly, this is intracellular, it's 10 nanomolar. So if one thinks about antibodies, the antibody would need to go into the neuron and capture it. I'm a bit more optimistic around antisense oligonucleotides, which would suppress all tau and the production of tau to actually capture also the high molecular weight fraction, which is mostly intracellular, not extracellular.
Thanks. We have a question from Ralph Kern. Can neuromodulation activate or rectify parvalbumin neuronal function and provide a physics-based approach to the treatment of Alzheimer's disease?
I think this is a very important and excellent question. Of course, I'm thinking of the work from Li Wai Tsai using 40 hertz gamma stimulation, which presumably has an effect on parvalbumin interneurons. I'm sure, I mean, this is not an area we explore ourselves, but I'm sure there's other ways of non-invasive stimulation that could potentially target parvalbumin interneurons as an active area of investigation. I think this is, for me, essential not only to think about molecular targets, but also to think about how can we restore these brain rhythms using, for example, non-invasive stimulation techniques.
A few more questions about target. So someone asks, what about oligodendrocytes as a target?
I think, again, the data we have supports an important role of oligodendrocytes in contributing to hyperactivity. I think your data is interesting that oligodendrocytes in Alzheimer's tissues are actually increased rather than reduced. So one could think that if neurons are lost and there are more oligodendrocytes, perhaps the contribution of oligodendrocytes to Abeta production as the disease continues, that the oligodendrocytes become more important. The question is how to target it. In these experiments, we block base 1. So can one block base 1 in oligodendrocytes specifically to reduce Abeta? Could be one approach. A cell type specific targeting so that we don't interfere with the neuronal targets of base 1, such as seizure-6, which seem to be not present in oligodendrocytes. Could one leverage these new alternative pathway that Mina Rüthen's group has discovered? I think these are all excellent ideas and things we need to pursue and to test.
So we've already gone over time, but I think we'll go through a couple more questions because there's still lots in the Q&A box. So Thomas Back asks, given the role of lower activity of inhibitory transmission that you delineated, wouldn't we expect a therapeutic effect of GABAergic medication, which is already being used for behavioral problems in dementia patients, but would it influence cognition?
So can you repeat the question? So is the question whether GABAergic inhibition or drugs that increase GABAergic inhibition should be beneficial?
Yes, I think so.
Yeah.
So given, yeah.
I mean, actually, experimentally, we have shown that enhancing GABAergic inhibition, for example, restores um, slow wave oscillations. What's critical is also the timing. If and this was done in a behavioral context. So if the so if the benzodiazepine in this case, which is not a good drug for Alzheimer's disease for various reasons, but as a test compound here. So if the benzodiazepine was given to the mice before the cognitive task, this was a water maze, it interfered with the water maze. But when we gave it directly after the learning task to enhance slow wave oscillations, the mice remembered better, and the next time they performed better. So I think these compounds are just not good enough. The existing compounds are just not good enough and have too many side effects. But the principle remains that I think GABAergic inhibition is an important target, and strengthening pathway-driven interneurons is important, but the drugs we have are not good enough and certainly shouldn't be given to people with Alzheimer's disease. There's actually literature that it can increase the risk of developing cognitive impairment and dementia. But I think this is not because, or I think this is more related to the problematic effects that these drugs have.
So we still have quite a lot of questions left, but we have gone 5 minutes over time, so I think we'll leave it there. So we can get to all the questions. Thank you everyone for attending, and I hope you enjoyed the talk. And thanks, Mark, for a great talk.
Thank you very much.